Oh, really?
I, s- I think I did stop the recording.
You hit the.
Hello, good afternoon, and welcome to the North American Vascular Biology Organization's third webinar in our webinar series. I'm Linda Shapiro from the University of Connecticut, and I'll be moderating today's webinar. We're pleased to welcome our speaker, Dr. Christopher Hughes, who will present his work entitled A Perfused Blood-Brain Barrier on a Chip. Dr. Hughes was not able to present his talk at Vascular Biology 2017 in Monterey this past October, so we're very happy to have the opportunity to offer you a longer and more in-depth presentation than he would have been able to give at the meeting. Before we get started, I wanted to go over some logistical aspects. Throughout the webinar, you'll be able to switch between the phone audio and the computer audio in case you have a problem. You can see this information on the audio section of the GoToWebinar control panel. If you experience technical problems, please click on the Help tab at the top of the control panel. Scroll to the bottom of the Help screen for the technical support phone number. At this time, I'd also like to welcome Duck Fan, also from the University of California, Irvine, who will monitor today's questions. Questions will be handled in two ways. Unfortunately, we're not able to interrupt and answer your questions during the presentation, but throughout the presentation, you can type your questions into the question box on the control panel. Those questions will be answered at the end of the presentation. At the end of the question and answer period, provided there's time, the attendees will be asked if there are any additional questions, and if you have a question, please raise your hand by clicking on the hand icon on the left-hand side of your control panel. You'll be recognized by the moderator and your mic will be unmuted, and you'll be then able to ask your question live. The webinar is being recorded and archived on the NAVBO website for future use. Dr. Hughes is a professor and chair of the Department of Molecular Biology and Biochemistry and a professor in the Department of Biomedical Engineering at the University of California, Irvine. He's also the interim director, Edwards Life Science Center for Advanced Cardiovascular Technology. His lab's primary interests are molecular mechanisms of angiogenesis, including the role of EMT transcription factors, slug and snail, development of microphysiological systems, body on a chip, for high-throughput drug screening, and molecular mechanisms of hereditary hemorrhagic telangiectasia, HHT, excuse me. Please welcome Dr. Hughes.
Oh, hello everybody. I hope you can hear me. Sorry about that. We had a little technical glitch while we worked out the microphones. So good morning to you, to those of you on the West Coast, like myself. Good afternoon to people elsewhere. And thank you for that introduction. So I'm going to be talking to you today about work that we've been doing in the organ-on-chip space, and particularly in our development of vascularized microtissues. And I'm going to talk at the end of this about the blood-brain barrier on a chip model that we have developed. I have to put in this disclosure to tell you that myself and some of my colleagues have formed a startup company called Kino Biosciences that is commercializing some of the technology that I'm telling you about today. So the brain, as we're all aware, is a highly complex organ. It has a high metabolic rate, and as a consequence, it needs a constant supply of nutrients and oxygen. As a result of this, there's a tremendous amount of vasculature in the brain, as you can see here, and in this diagram here. Now, the brain is an extremely delicate tissue, as we're well aware, and there are classic experiments done many years ago injecting a dye called Evans blue into mice or rats, And this rapidly gets out of vessels and distributes through the body and basically turns the animal blue. Interestingly, if you open up the brain case, you'll find, as you can see here, that the brain remains pristine white. If you traumatize the brain in any way, then some of this dye will leak out into the brain tissue. And these kind of studies were the first indication that the vasculature in the brain was significantly different to vessels elsewhere, in that it was not freely permeable to many molecules. And this came to be known as the blood-brain barrier. And so there's a lot of interest in the blood-brain barrier in a basic sense of how does it work, and what is the mechanical, what are the mechanical properties of the blood-brain barrier. But also it's extremely important in pharmacology because a lot of drugs that get into most tissues don't get easily into the brain because of the blood-brain barrier. And so we're all very interested in understanding the blood-brain barrier and how to get drugs across it. So this is what the blood-brain barrier is comprised of. It used to be thought of as being purely the endothelial cells that line the vessels in the brain, but we now know that it's a lot more complex than that. So in red are the endothelial cells. These are distinctly different, as we'll see, to endothelial cells elsewhere in the body. One problem with this diagram is that there's actually a tight seal between the endothelial cells at various junctions, but particularly tight junctions. So that forms the first line of the barrier. There's then a basal lamina that wraps around the endothelial cells. This also, we now know, contributes to barrier properties. And then on the outside of this, and sometimes embedded within this, are perivascular cells called pericytes. These contribute to barrier function by crosstalk with the endothelial cells, and we'll see some data on that in a moment. And then there are astrocytes that put down foot processes onto the basement membrane and the endothelial cells. And this combination of pericytes and astrocytes helps to maintain barrier properties of these endothelial cells. And if you put these endothelial cells somewhere else in the body, where there are no astrocytes, for example, then they will lose the barrier properties. Likewise, if you put tissue, if you allow vessels to grow into transplanted brain tissue anywhere else in the body, then those vessels that enter the brain tissue will take on blood-brain barrier characteristics. So our work on body on a chip began when DARPA, a big government agency that funds a lot of the crazy research, decided that they wanted to do something would really push the field forward, and that was to create ideally a palm-sized device that would have at least 10 fully functional human organs, and that this could be used to test for drugs or environmental toxins. And to give you a sense of how DARPA thinks, the director of this program, Barry Pallotta, told us in our first meeting, if 90% of what we fund doesn't fail, we're not doing our job. So they really don't like funding incremental work. They go for moonshots. And so this was their idea. So they funded part of this initiative, and then NIH funded part of the initiative. And myself and colleagues were funded to develop a heart on a chip and a tumor on a chip. And along the way, we started to develop other organs as well. So why did we want to go through all this effort to put miniaturized organs onto chips? So the reason is that organs are complex tissues. They're three-dimensional, unlike a lot of what we do on dishes, which is two-dimensional. And there's a lot of complexity. So this is a diagram of a tumor. And within there, you can see all the different cell types. There's the normal cells. They're the tumor cells that derive from those. But then there are a lot of other cells as well. There's vasculature, there's lymphatics, there are fibroblasts, there are support cells around the vasculature, such as the pericytes. And then quite often there's an immune component in there as well. There can be lymphocytes, macrophages, etc. So a highly complex and three-dimensional organ. At the bottom left, you'll see an H&E stain with some staining there, actually not an H&E, an immunohistochemical stain for vasculature, which shows up as brown. And this is to emphasize that in this tumor tissue, there is vasculature. And in fact, in all organs that you look at, there is vasculature. And so this is where we've been, two-dimensional cultures, so often monocultures, so a single cell type growing as a monolayer in a plastic dish. And it doesn't take much to realize that this is a very poor model for what you see on the top left. It's not three-dimensional, there aren't multiple cell types in there, and a plastic dish is thousands of times stiffer than a normal tissue. So there has been some movement towards spheroids, and in the bottom right in A and B, you'll see some spheroid type cultures there. These are three-dimensional. People are starting to put different cell types in there, but they still lack the vasculature, and we know that that's important in all tissues for delivering oxygen and nutrients. And there are some cells that just won't form spheroids, as we'll see. When cells form spheroids, they do it because they're expressing adhesion molecules that allow the cells to stick together. And as we'll see, there are cells of interest that have lost these adhesion molecules and no longer form spheroids. So what we felt we needed to develop were three-dimensional tissues, human tissues, because we understand that while mouse is a good model for many things, it's not a great model for a lot of what goes on in the body, perhaps not surprisingly. And then, as I said, all tissues have a vasculature. And so if we really want to model how three-dimensional tissues work, then we should have vascular tissue in there as well. And if we're going to put blood vessels in, then we may as well have them perfused. So that was our goal in this project. So this is just to point out why it's important to look at drugs in three-dimensional tissues. So you're looking at dose response curves here in a three-dimensional assay. It doesn't matter what it is, but for those who know our work, this was one of our bead assays, a sprouting assay for vasculature. And these are 3 FDA-approved drugs. And just focus on the red dotted lines. I've put those in at roughly the IC90, so where we've blocked about 90% of what we're interested in, which in this case is sprouting. If we test these drugs in a 2D assay, and this can be proliferation or migration, then if you look at those dotted lines, that's where the IC90 was in 3D, and you can see these drugs have no effect in 2D. And we've got some positive controls over there on the right to show that we can block these functions. The only reason we have these drugs is because they were targeted, they were designed to block VEGFR2. If you had done a normal 2D screen, you would never have pulled these drugs out of a library, which suggests that many of the drug libraries that we have contain really interesting drugs that we don't know about because we're using the wrong assays to look for them. So what we decided to do was to create vascularized microorgans. So here's a spectrum of assays that one can use. And on the left are the 2D monolayers we're familiar with. These are obviously very simple, but not terribly physiologic. Where we'd like to be is over on the right, the humans, obviously physiologic, but clearly not simple. We're talking large-scale clinical trials there. There's a lot of work has been done on mouse. It's physiologic, but in many ways differs from human. For example, as we all sit here, our hearts are beating somewhere around 70 beats per minute. If you had a mouse sitting on your desk, its heart would be beating about 300 beats per minute. If you got up and ran downstairs and came back up, your heart rate would be up to maybe 150. The mouse, if it did something similar, would be up over 800 beats per minute. So is the mouse a good model for looking at drugs that target the human heart? Probably not. Liver also is very different. So we wanted to stay away from mouse tissues. I mentioned spheroids are quite good, but have some drawbacks. So what we've created in the middle there are these three-dimensional vascularized microorgans. In red is the vasculature. So there's an artery at the top, a vein at the bottom. Those are actually microfluidic channels that are representing those vessels. And we form a living capillary network that connects the artery to the vein. And then we can flow a blood substitute through these vessels. And that will provide nutrients to any cells that we put in there with the vessels. In this case, these green cells are tumor cells. So I give this talk to people who aren't vascular biologists. I'm guessing because this is NAVBO, most of you are. So you will recognize this, the high pressure arterial side of the vasculature, the low pressure venular side, and that's connected by a capillary network. And it's through the capillaries that oxygen and nutrients are delivered to tissues. So we schematize it this way. And so there's that diamond is the tissue chamber, and we form the vasculature in there. And then medium is run through these vessels driven by gravity. So in this diagram, you'll see three little diamonds in the middle of this platform. Those are the diamonds that you see at the top there and the bottom left of our screen. That's where the tissue goes. And then the medium is driven by gravity. So because V1 is higher than V2, medium will flow through that channel, and that represents the arterial side, or the red vessel in the bottom left. Because V3 is higher than V4, medium will flow from V3 to V4 through the vein. And because V1 is higher than V3 and V2 is higher than V4, the net flow is from V1 through the vascular network to V4. And we can control the height of medium in each of these tubes. And by doing so, we can very carefully control the flow of medium, the flow rate and the shear rate through these vessels. We always do console simulations before we do redesigns and make new platforms. And this allows us to test what rates of flow we can achieve in each platform and what column heights we need to get the flow we need. And here is one of those vascular networks. The endothelial cells are transduced with the lentivirus so that they express fluorescent proteins. In this case, they're expressing GFP. The picture above, they're expressing M-cherry. And you can see that these form nice tubes, and these are all interconnected, allowing flow. And we get anastomosis of in this case, the artery at the top to the capillaries. And you can see on the bottom and the right that these are indeed tubes. And the dimensions of the chamber is about 2 by 1 millimeter, and it's only about 100 microns thick. We can put in multiple types of endothelial cells. We have stromal cells, which are required for generation of the vasculature. Those stromal cells, we've published on those before, and they make a lot of matrix molecules. We include matrix, but that gets rapidly remodeled by the stroma. And the matrix we start with has some fibrin, and then any extracellular matrix from any tissue that we're interested in, we can put that in there as well. So here you can see we've actually labeled the stromal cells in this case. We normally don't, but you can see those in red in the background. In this case, the vasculature is in green. Here we've put in 70 kilodalton rhodamine dextrans, so this is about the size of albumin. And you can see that red dye is flowing into the green vessels. And this particular one is coming out the bottom left as opposed to the bottom right. These days, we always have it coming out the bottom right. And we can put in fluorescent beads, and you can see that those flow through these vessels. And again, we can control the rate of flow of these just by dialing in different heights of the column of the medium. And we've also put in blood. So here's red blood cells, and you can see that this looks very much like you would see in skin vasculature, for example, where you get areas of slow flow in the bottom right, and then accelerated flow across the middle there. And you can see at the bottom, some of these vessels are so narrow that the red blood cells are actually having to be squeezed through these vessels. And that's something you see in vivo, particularly in brain, where many of the capillaries are only about 5 microns in diameter, whereas a red blood cell is 7 microns in diameter. So this is how the vasculature develops over time. The cells go in as a slurry in the gel. You can see on day 0. And then by day 4, this vascular network has formed. And this is stable over several days, up to 21, sometimes up to 28 days. And as I said, all the tissue is put into this in a slurry of gel and cells. And then this vasculature will form spontaneously over the next few days. One of the things we were asked to do by NIH that seemed terribly obvious to us, but we did it anyway. They wanted us to cut off flow and see what happened. So in vivo, this is basically an ischemic attack. So if for some reason you get a blockage in a vessel, then there's no oxygen and nutrients going to tissues downstream of that blockage, and you would expect those tissues to die. So we set up tissues here, and at day 14 we stop the flow, and then you can see the tissues begin to die off, and after 6 days most of the cells are now dead because of a lack of nutrients. So this very much models what you see in vivo. People like to ask what endothelial cells can we use for this. We use a lot of ECFCs. These are cord blood derived endothelial cells, but Hughes work extremely well in the device as well, as do iPS-EC, as you can see there on the right. One of the key features of the capillary endothelial cells is that they recruit support cells, and in this case, pericytes. And so here we see the red vasculature, and these endothelial cells recruit the pericytes, and these pericytes wrap tightly around the vessels. So you can see these pericytes in yellow. Here again, the vessels are in red, the pericytes are in yellow, and we're running a green dye through the vessel there. And this is just to confirm that the support cells are indeed pericytes. They're all expressing NG2, and we've looked at lots of other markers as well, such as PDGFR beta. And in combination, the endothelial cells and the pericytes cooperate to lay down basement membrane. And we've seen beautiful data before from George Davis's group showing how each of these cells contributes. And we've just stained for collagen 4 here to demonstrate that a basement membrane is rapidly laid down between the endothelial cells and the pericytes. So the top left here's a little movie that's going to run, and you're going to see a green dye running through these vessels. So everything that we've seen about these vessels makes them look like they are operating as vessels do in vivo. But to confirm that, we wanted to check their permeability. So as I showed you before, we can put through 70 kilodalton fitzy-dextran. Again, this is about the size of albumin. And you can see that over time, this slowly leaks out of the vessels. And we can calculate a permeability coefficient. And this matches very nicely to what we know about capillaries in vivo. For example, skin capillaries have a similar permeability. We then challenge these vessels with thrombin, and thrombin is known to induce vascular leak. And you can see here that the vessels respond rapidly and become extremely leaky. So they do indeed respond to challenge. And then we also put in something the size of immunoglobulin, so 150 kilodalton fitzy-dextran. And again, you would expect this to leak out, but leak out more slowly than the smaller molecule. And that indeed is the case. You can see there's only a very slow leak from these vessels. And as you would expect, the permeability coefficient is substantially lower. So we were interested in the recruitment of parasites. And so here, under flow, you can see the red endothelial cells and the tight apposition of the yellow parasites. And so we wondered what would happen if there was no flow. And so early, if we stop flow, this is before anything dies, we see that we don't get recruitment of the parasites. And what we hypothesized here is that under flow, the endothelial cells are induced to express PDGFB, and that then diffuses away from the vessels, binds to PDGFR beta on the parasites, and induces recruitment of those parasites to the vessels. And so under flow, we added an inhibitor of the PDGF receptor, and that not only blocked recruitment of the parasites to the vessels, but it also led to an increase in permeability of the vessels. And we need to do some repeats of this, obviously, to get these arrow bars smaller. But what this suggests is that the parasites do contribute to the ability of the endothelial cells to mature and reduce their leakiness. So put it another way, the parasites in some sense control the permeability of the endothelial cells. So we wanted to use this model in our sort of basic research, and an opportunity came up to do that. We've been interested in the transcription factor slug and how that regulates angiogenesis. And so we overexpressed slug in endothelial cells, and we looked to see what genes were regulated. We saw all sorts of interesting stuff, but one of the things we noted was that when slug is turned on and it induces angiogenesis, the endothelial cells downregulate adhesion molecules and junctional molecules. Here we just show confirmation, show the actual data for occludin, and you can see when we overexpress slug, we get a reduction in occludin expression. We also see reduced protein expression of other junctional proteins such as VE cadherin, and you can see that in the stain at the bottom. So we predicted that if we overexpress slug in vessels, then they may become more permeable. And so we generated control vessels. There's some on the left there. We put through 70 kilodalton fitzy-dextran. You can see there's very little leak there. We then formed vessels with slug overexpressing EC, and you can see firstly that this leads to some very strange looking vessels. The vessels do form, they do carry flow, but they're massively leaky, as you can see here. So that was a nice way to use the model to confirm that loss of junctions in endothelial cells does indeed have consequences for permeability, as one would predict. So we next looked at tumor growth in the platform. And so here we've got vessels in red, tumor cells in green. Remember that there are stromal cells here, and parasites, etc., which we're not seeing. And this is just a picture to show you that they are there. So the endothelial cells in this image are green, and the parasites are in red. You can see those wrapping around, and then the tumor there in blue. And remember, all the nutrients that the tumors are receiving here, and any drugs we give, they are receiving them through these living blood vessels, just as they would in vivo. So you can see here that the vasculature does go in and around the tumors, as again you would see in vivo. And as you might expect, this is a nice platform for looking at tumor angiogenesis. So here you can see the tumor in green and vasculature around it. And then over time, the vessels are recruited to this tumor. So by day 6, you can see that the vessels are now fully in and around the tumor. providing nutrients, and the growth of this tumor really takes off at this point. We have looked at drug studies in this platform, and the standard of care for colon cancer is Folfox. And you can see in the top there how these tumor cells, they grow as clumps in the device, and you can see those growing over time. In the presence of Folfox, you can see that we can control very nicely the growth of this tumor, and in fact we can even see some reduction in tumor size after we add the drug. We've also looked at a triple negative breast cancer. Now this is one of those lines that will not form spheroids. The cells have lost a lot of adhesion molecules, which is why they're so invasive in vivo. And it's also why they won't form spheroids. However, in the device, you can see again, these cells recapitulate what we see in vivo. They're highly invasive. The standard of care is Taxol. And again, we can control the growth of this tumor with Taxol. And Taxol is also known to be a vascular disruptor, and you can see that happening in this tumor as well. We've put in a glioblastoma. This is a highly aggressive tumor. You can see how quickly this is growing. Standard of care is temozolomide, and you can see that at the bottom is controlling tumor growth. And there's a new drug we're testing here at UCI, and that is highly effective at controlling the growth of this tumor. And you can see those data here. So we've tried multiple tumor types, as you can see. We've tried colon, glioblastoma, various breast cancers. We've also tried melanoma. Everything we've tried so far grows nicely. And you can see here the different ways that these tumor cells grow. So the SW620s, they grow as tight knots of cells. The MDAs are highly invasive. And the glioblastoma is also invasive, but has yet another phenotype. We've noted that the tumors are quite often more resistant to some drugs in 3D than 2D, and you can see that here. The curve shifts strongly to the right in 3D. However, we also see drugs go the other way. So again, on the left here, the drugs are more effective in our 3D cultures than they are in monolayers. On the other hand, we've got drugs such as tamoxifen, sorafenib, gemcitabine. These are more effective in 2D than they are in 3D. So it's pointing out again the importance of testing drugs in a proper environment. 2D monolayers can give you data that's not going to be representative of what actually happens in vivo. We can also see that some drugs differentially affect tumor or vessels. So, for example, pazopanib targets VEGFR2, and that strongly disrupts the vasculature. 5-FU very much targets the tumor rather than the vessels. Sorafenib targets both. And of course, if you target the vessels, then the tumor will eventually suffer because of a lack of nutrient delivery. We have imaged metabolism in tumors. It's a lot of interest in controlling tumor metabolism these days as a way of controlling tumor growth. We use a process called fluorescence lifetime imaging microscopy. And just in the interest of time, the white to blue is more glycolytic and the red is less glycolytic. And so what you're seeing here is that the endothelial cells are more glycolytic than the pericytes. If we put in tumors and look again, and if you just focus on the middle two panels there, this is a relative scale. So now we see that the tumor is highly glycolytic, whereas the vessels and the stroma are less glycolytic than the tumor. So you can think of it as a spectrum, that the tumor is most glycolytic, then the endothelial cells, then the stroma. And if we put in drugs, we can manipulate the tumor while leaving the vasculature intact. And if you just look at the graph at the bottom right, doesn't matter what the axes are, the point is that you can see a shift in the metabolism of the tumor when you add drug. That's the dots, whereas the metabolism of the vessels, the triangles, is not altered. So this suggests there's a window whereby we can target tumor metabolism while not affecting metabolism of the stroma and the vessels. So this is looking at tumor heterogeneity. This is a lot of interest in this now. And you can see there are areas within the tumor that are more glycolytic than other areas. And again, you can see this here. There are areas, sort of hot spots, if you like, of metabolism. We did a comparison between cells growing in vivo, and those are the orange triangles, and those same cells growing in the vascularized microtumor. Just ignore the blue dots, that was a treatment in the mouse. And you can see that there's very strong overlap in the size of these dots. So if we just go back a slide here, if you see the heterogeneity in these tumors, you can see sort of dots of highly glycolytic activity. If you measure the size of these and then the interspot distance, that's what's being measured here. And again, you can see strong overlap between what we see in our vascularized microtumor and what you see in vivo. So in the last 10 minutes or so before we open up to questions, I'm just going to give you some background, a little more background and some data on the blood-brain barrier model that we've developed. So we're very interested in taking our vasculature and trying to create a blood-brain barrier. So just as a reminder, these are the cells that are involved, the endothelial cells, the perivascular cells or the pericytes, and the astrocytes. And so we've put all of these into our device. So if we start at the top two panels, the endothelial cells there are green and the astrocytes in red, and you can see the astrocytes pushing out these processes, and these processes are sticking down to the endothelial cells just as they do on that image on the left. So they are wrapping their foot processes around the endothelial cells. And then the bottom left, we have the endothelial cells in red, the astrocytes are now in blue, and again you can see those reaching out to the vasculature, and then the pericytes in green wrapping nicely around. So we're very much recreating the structure that you're seeing on the left, and the final component of that is the collagen IV basement membrane at the bottom left. You can see that. And here we've actually stained an astrocyte for GFAP. It's a good astrocyte marker. And again, you can see the cell reaching out to the endothelium. So we've looked at tight junctions. We see Z01, there's CD31 as part of the junctional complex there. We've also looked at Claudin 5. This is a very diagnostic marker of blood-brain barrier endothelium. And again, you can see strong expression of that, and you can see those junctions outlined there on the bottom right. Some higher magnification images. Again, you can see the extensive collagen IV basement membrane that's been laid down around these endothelial cells. And again, in blue, you can see the astrocytes reaching out. And so we've done some reconstructions, so you can see these in 3 dimensions. Here, the green is actually labeling the pericytes, and you can see how tightly those are opposing the endothelium. And you can see in some cases they're just extending out filopodia and processes, and then these are wrapping around the endothelial cells. In other cases, you can see the whole cell body is tightly opposed to the endothelium, basically adhering to that collagen IV basement membrane. And just again here, on this confocal Z-stack, you can see that there's a nice red lumen of the endothelial cell, and then the green pericyte wrapping around that. So we wanted to look at function. Firstly, we looked again, as I said, at gene expression. You can see in the presence of astrocytes, we see an upregulation of junctional molecules like V-ecodierin, CD31, and Claudin 5. A couple of blood-brain barrier markers, so the taurine transporter and GLUT1. And again, in the presence of astrocytes, you can see a strong upregulation of those. And then we also looked at some others. So occludin, as I've shown you, is an important tight junctional protein. We looked at gene expression in the whole neurovascular unit here. So this is all of the cells together, but many of these are endothelial markers. And for example, SLC2A1, that's GLUT1, strongly upregulated. And two transporters that are important for the efflux of compounds from the brain. It's another way that the brain keeps unwanted compounds out, is that it has things like P-glycoprotein and MRP4 that will rapidly pump drugs back across the endothelium, back into the blood. And again, you can see both of those are nicely upregulated in this model. And once again, there's some junctional staining there of these vessels. So we've done modeling of flow in these vessels. Again, you know, it's very important to get the flow rate and the shear rates correct. So what we can do now is we import pictures of actual networks into this modeling software, and then we do computational fluid dynamics to model what the flow rate is. And you can see the acceleration of flow there in some areas and the increase in shear in those areas. And then again here, you can see how the flow moves through these vessels, so that this allows us to calculate flow rates and shear rates. A classic thing, and I showed you right at the beginning with blood-brain barrier, is to put Evans blue into an animal for a few minutes and then flush it out. And you see that Evans blue leaks into most of the tissues of the animal and turns it blue, but doesn't leave the brain vasculature, and so leaves the brain white. So just for kicks, we ran Evans blue through our vessels, and as you can see, it doesn't leak, which is very good. We do know that the astrocytes contribute to the tightness of the barrier. Here on the left, you can see in the absence of astrocytes over time, we get significant leakage of 70 kilodalton fitzy-dextran out of the vessels, whereas in the presence of astrocytes, these vessels are much tighter, and we don't see much in the way of leak. And so we've now quantified this, and in the squares, that top line, this is the leak over time from our regular vessels. And then the bottom line, the black dots, is the leak through the blood-brain barrier vessels. And you can see that there's significantly less leakage through the blood-brain barrier vessels. We estimate in different preparations anything from 4 to 10 times tighter vessels in our blood-brain barrier than normal. So we're now looking at drug delivery across this. We're looking for ways to measure drug delivery into the brain tissue. We're looking for treatments that might tighten barrier under disease processes. And also we're very interested in glioblastoma. There are data out there that suggests this causes a leak of the barrier, but it's still hard to treat because it's invasive. And where it's invasive, it doesn't necessarily traumatize the barrier. And so we're going to now use this model to try and identify drugs that can cross the blood-brain barrier and target glioma. So this is pretty new data I'm showing you here. This is just a D54 glioma in the VMT. We haven't yet done the permeability studies with this. So to summarize what we've talked about today, tissue and tumor microenvironments are complex, and the only way we're going to really understand these is to use three-dimensional models that capture the complexity of real tissues. We've looked at vascularized microtumors, and this allows us to study the crosstalk between the various cells that are in a tumor. We've noted significant differences in drug responses of tumor cells when we assay them in three-dimensional culture versus standard 2D cultures. And we've now been able to model multiple different types of tissue in our device, including colon and tumors, brain tissue, blood-brain barriers I've shown you. We're doing diabetes studies by putting islets in here. A colleague, Steve George, is developing a bone marrow model with putting cardiomyocytes, and we're also developing a liver model. And so in the final slide is always the most important one. It's my lab and the people that have done this work. They're all listed out on the left. Every one of those has made tremendous contributions to this work. I particularly want to call out Duke Fan, who's with me here today. He is our microfluidics guru, and without him, we'd have made very little progress on this project. This has all been a collaboration with Steve George, who used to be here at UCI, who's now at UC Davis, and Abe Lee, who is still here at UCI. Our labs have been working on this together for 10 years now. Micah Sanders, a colleague I'm collaborating with in San Diego on some of this work. Rosemary Ackhurst in San Francisco, we've been collaborating with. And a big call out to NIH, who've generously funded this work for the last several years. And with that, I'd be very happy to take questions.
Okay, well, thank you, Dr. Hughes. That was great. I see we have several questions from the audience, and I'm going to turn this over to Dr. Fan so he can ask Dr. Hughes the questions.
So we have a few questions from Dr. Bill Mueller. The first question is the formation of the capillaries network affected by having flow around? That is, if you start the network forming while fluid is flowing through the diamond-shaped chamber, does it affect the rate of formation or characteristic of the capillaries network?
That's a great question, Bill. Thank you very much for that. That was some of the earliest work we did, actually. We found, and we did publish on this, that the interstitial flow through the tissue is actually a critical component in the initial formation of the network. And we did some modeling on this, and it seems that it's a contribution of both the shear that the endothelial cells experience, but also distribution of growth factor across the device. So in the absence of flow, the formation of the vasculature is not good. So we do need that interstitial flow to get the initial generation of the network. Once the network has formed and anastomosed the outer channels, then the shear that the endothelial cells sense is definitely a component in remodeling of the vasculature. So when we have low flow rates, the vasculature looks one way, and then if we increase the flow and the shear, the vasculature becomes a lot more regular looking. We don't really have a good way of describing it. It's something you get used to seeing, but it looks more organized, a little more hierarchical once you have flow through there, and once you hit that critical shear threshold of 5 to 7 dynes per centimeter squared when you turn KLF2 on, and then you definitely see remodeling of the vasculature under flow.
So the next question also from Dr. Bill Mueller is, have you tried to put leukocyte through your system, and would you be interested?
Yes, we've only done that so far as a proof of concept. So we did take some human monocytes and we labeled those, and we put them into, if you recall the design, into V1, so the top left. So they flow then through the artery into the network. And we saw a rest, and we saw extravasation of those cells into the surrounding tissue. So we are very interested in pursuing those studies. We haven't yet put lymphocytes in, but we do have a plan to do that. Our model is allogeneic, it's not autologous. And so there could be some interesting things if we put allogeneic lymphocytes in, because as some of you know, my early career in vascular biology was looking at antigen presentation by endothelial cells, something that only happens in human. And so if you put in allogeneic T cells, And if there's a little bit of interferon around and you get upregulation of MHC on the endothelium, then you can get activation of those T cells. So these are all studies we would like to do at some point, but the short answer is yes, leukocytes, we can flow them through, and yes, they do adhere, and yes, they do extravasate into the surrounding tissue.
So we have two questions from Kishore Wary. The first question is, were you able to use iPS-derived endothelial cell to form the blood-brain barrier? And the second question is, besides Hughes, did you use any other primary endothelial cell to make the blood-brain barrier?
So we have not tried IPS for blood-brain barrier yet. That is something on our list of things to do. The cells that we've used so far are the cord blood-derived cells, and we induce those to take on a blood-brain barrier phenotype. So as an aside, and I alluded to this in the talk, there was some beautiful work done, I think in the 80s, with quail chick chimeras. And so quail, I think, have oval-shaped nuclei and the chick has round nuclei. So you can tell where each tissue is from when you make chimeras. And what was found there is that if you transplant brain tissue anywhere into the organism, then the vessels that grow into that take on a blood-brain barrier phenotype. So what that tells you is that the blood-brain characteristics of the endothelial cells is not something that's inherent so much in their lineage as inducible by their environment. So what we did is to take regular endothelial cells, and these ECFCs are pretty plastic, which is helpful, and then put them in a brain environment. And as you can see, they take on blood-brain barrier characteristics. We have tried commercially available brain endothelial cells. They don't make great networks. We suspect that they've been passaged a little too many times before we get them, which has been a problem with commercial cells. We use ours at very low passage. So yes, we have tried other endothelial cells, but as of now, our ECFCs are working very nicely. So the next step is to do it with IPS, though. We are keen to do that.
So we have two questions from Taliha. The first question is, which parasite was used to construct the blood-brain barrier? And then the second question from Taliha is, do astrocytes interact with parasite?
Huh, interesting. The parasites, I'm probably actually going to defer this one to Duke. I think they are all placental derived, is that right? So the parasites we've used are placenta derived. We haven't done it with brain derived yet. Do the astrocytes interact with the parasites? I don't know. I guess we could look more carefully in our confocal images. What we've been focusing on is how each cell interacts with the endothelium, but I can't say I've ever specifically looked to see if there are interactions between the astrocytes and the parasites. So it's a great question. I don't know. And I don't even know in the brain whether there are interactions. So if anyone knows, maybe they could type that in. And but no, I don't know the answer to that one.
The next question comes from Jerome Robert. How could the lymphatic system be implanted in your system?
Yeah, a lot of people have asked us, have we done lymphatics? And the simple answer is no, we haven't. One of our issues in the lab, and I'm sure Duke will attest to this, is a lack of bandwidth. There's a lot of cool stuff that we could do and would like to do that we just haven't got around to, and lymphatics is one of those. I suspect it would work, and we do have a little data suggesting that under some conditions, IPS-derived endothelial cells can take on lymphatic phenotypes. And we do know that those work in the device. So my guess is we could do it. We just haven't yet.
So the next question is from Douglas Marchuk.
Oh, hey, Doug.
Do the EC on the low flow side of the chamber take on expression of Venus marker? And conversely, do the cell in the high flow side take the expressions of arterial marker?
That's a great question, Doug, and it's something that we just haven't done yet. I would like to think that that happens under the high flow in the arterial side and the low flow on the venular side. There are some technical reasons that we haven't done that to date, but we have a new design of the device that you may have seen me present that at one of the HHT meetings. I'm not sure if you've been at one of the recent ones, but it's a redesign that has allowed us to generate AVMs, what we think are AVMs in the device, suggesting that there may actually be an arterial and a venous side. The other thing that we could do, and again, haven't done yet, is to put arterial cells in one side and venular cells in the other side, and then form a capillary network between them. So we would force it to be arterial on one side and venular on the other. Again, it's a cute experiment to do, but we just haven't done it. But those are the kind of things we're going to be looking at. We've got an HHT project, and I just noticed, you're not seeing it, I'm looking at my final slide, and I don't actually have my new HHT funding listed there, but we are now supported by NIH to do an HHT project, and the questions that Doug has raised are some of those that we will be addressing in that project.
So Dr. Bill Muller has a follow-up answer. When we put astrocyte conditioned medium into culture with HUVEC, they start expressing Claudin 1 and Claudin 5 and make really tight junctions.
Oh, wow.
Brain capillaries, endothelial cell line under the same condition don't work better. In fact, they are worse.
Oh, thanks, Bill. Yeah, I can't say that surprises me. As I said, you know, we have evidence, and there's those beautiful studies in the 80s suggesting that blood-brain barrier is an inducible phenotype. So that's really good to hear that. I'm also fascinated that conditioned medium works. That actually is a surprise to me. I was figuring it would have to be a contact-dependent thing, but that's really interesting. So next
Hey,
time
Chris. Oh, sorry. I think Doug Marchuk had another question or a follow-up for you too.
Oh, okay.
Doug, you're unmuted.
Oh, no, I didn't. Did I do something? I must have pressed a button with my,
Okay.
but I didn't mean anything. I was typing them. I didn't know I was sending messages. Can you hear me?
Okay.
Oh, okay, yeah. Hi, Chris. But yeah, sorry. No more questions.
We have another question from Leah Zhong. Just curious, are the vessel grown with the tumor cell more permeable?
Yeah, I've been asking the guys in the lab to do that experiment for probably close to a year now, and I think they're actually doing it. Is that?
Yeah.
Duke is nodding his head. So those experiments are being done. I'm desperately keen to know the answer to that as well. I did see some preliminary data at one point that suggested that they were leaky, but yeah, I like you, I don't know the answer to that yet, but it sounds like we will have an answer in a week or so, which is good.
I think we have Thomas Newman would like to connect.
Oh,
and here's a question from Thomas Newman. How have you run the system, and why does it appear collapsing at day 21?
how long have we run the system? Hi, Thomas. Nice to have you on here. We have taken them out to 28 days before, which was a requirement for the NIH award we had. The issue that we have is overgrowth of the stromal cells. So we have to have stromal cells in there. They basically condition the matrix. They pump out a lot of matrix molecules. We published a couple of papers on this a few years ago from Andrew Newman in my lab. And so we have to have them in there to help the endothelial cells make these networks. but they continue to divide and migrate, and they eventually overwhelm and degrade the matrix. So we're currently looking at some strategies to calm down the fibroblasts so that they stop doing that. Once we can get them quiet and quiescent, then I don't see any reason that the platform shouldn't run for weeks. But that's the limiting factor right now. It's the stromal cells.
Okay, this is Linda Shapiro again. Thank you both, and I see we're out of time. If you have any more questions, please email us at info@navbo.org, and we'll make sure you share your questions with Dr. Hughes and Fahn. I hope you enjoyed this as much as I did and found this information beneficial. Please join us again for our next webinar on June 14th, when Juan Malero Martin of Boston Children's Hospital will present his research entitled Engraftment of Bioengineered Vascular Networks. Just a note, please fill out the evaluation form at the end of the webinar or when you receive it in a subsequent email, and let us know what you thought of the webinar and any topics you might want to see on a NAVBO presentation. Based on responses from our earlier webinars, our August webinar will be on lymphatics, presented by Michael Dillinger of UT Southwestern. Thank you, and see you in a month.
Thank you, everybody.